Next, we'll discuss about the C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway. Though these plants have the C4, oxaloacetic acid is the first carbon dioxide fixation product they use in the C3 pathway. What are the characters? They have a special type of leaf anatomy. They tolerate higher temperature. They show response to the high light intensities. They lack process called photorespiration and have great productivity of biomass. Anatomy of C4 plants differ from C3 by having large cells around the vascular bundles, which are called bundle sheath cells, and the leaves which have such anatomy are known as Kranz Anatomy. The bundle sheath cells may form several layers around the vascular bundles and have large number of chloroplast thick walls impervious to gaseous exchange and no intercellular spaces. The pathway followed by C4 plants are called Hatch and Slack pathway. It is a cyclic process. The primary carbon dioxide acceptor is a 3-carbon molecule phosphoenol pyruvate and is present in the mesophyll cells. The enzyme responsible for the fixation is phosphoenol pyruvate carboxylase. It is important to register that the mesophyll cells lack Rubisco enzyme. The C4 acidase oxaloacetic acid is formed in the mesophyll cells. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release carbon dioxide and a 3-carbon molecule. The 3-carbon molecule is transported back to mesophyll where it is converted to PEP again, and thus completing the cycle. The carbon dioxide released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells enrich in an enzyme ribulose biphosphate carboxylase oxygenase, that is short form Rubisco, but lack in the PEP case. Thus, the basic pathway that results in the formation of the sugars, the Calvin pathway, is a common to the C3 and C4 plants. Check one more time. C4 Hatch and Slack pathway. C3 plants have oxaloacetic acid. It is a 4-carbon compound as the first carbon dioxide fixation product. Sugarcane and maize are the C3 plants. Have a special type of leaf anatomy. For the C4 plants is called Kranz Anatomy. C4 plants contain dimorphic chloroplast, tolerate higher temperature, lack a process called photorespiration, have a greater productivity of biomass. Now that the leaf anatomy, check in the picture. Next up, at the Kranz Anatomy, it is called the Wreath. Now, dimorphic chloroplast, C3 plants, non-cyclic photophosphorylation occurs in the granule thylakoids, during which oxygen gas is released as the byproduct of splitting of water. Agranal chloroplast occurs in bundle sheath cells of C4 plants. As these cells have stromal thylakoids only, non-cyclic photophosphorylation does not occur. As a result, oxygen gas is not released. Only cyclic photophosphorylation occurs in which ATP is formed. Now see the detailed anatomy of the C4 plants. Mesophyll cells have the PEP carboxylase for reaction of carbon dioxide and PEP to form a 4-carbon compound. Bundle sheath cells have the Rubisco for Calvin cycle. What are the comparison of anatomy of the C4 and C3 leaves? Check in the picture. Mesophyll cells are Rubisco for Calvin cycle. Bundle sheath cells have no Rubisco. These do not fix carbon dioxide. Mesophyll cells have the PEP carboxylase for reaction of PEP and carbon dioxide to form a 4-carbon compound. Bundle sheath cells are Rubisco for Calvin cycle. Close association permits carbon dioxide from mesophyll cells to bundle sheath cells for Calvin cycle. C4 pathway, see in the picture, the mesophyll cells, the enzyme PEP carboxylase adds carbon dioxide to PEP, phosphoenol pyruvate. A 4-carbon compound oxaloacetic acid conveys carbon dioxide into bundle sheath cells via plasmodesmata. In bundle sheath cells, carbon dioxide is released and enters into the Calvin cycle. What is a Kranz anatomy? Large cells around vascular bundles of C4 plants are called bundle sheath. Such anatomy is called Kranz anatomy. It means wreath, flowers arranged in a circle. The C4 plants have two types of photosynthetic cells, mesophyll cells and the bundle sheath cells. Presence of two types of cells allows separation of light reaction and carbon fixation. As a result, release of the oxygen takes place in mesophyll, while carbon dioxide fixation catalyzed by the rubisco occurs in bundle sheath cells. Thus, rubisco, which is present only in bundle sheath cells, does not come into the contact with oxygen. As a result, oxygenation activity of rubisco is completely avoided. To further reduce occurrence of photorespiration, C4 plants adopt a carbon dioxide concentration mechanism. This mechanism builds up carbon dioxide concentration in the vicinity of rubisco in the bundle sheath cells. Higher concentration of carbon dioxide near rubisco favors carboxylation and suppresses photorespiration. In C4 pathway, carbon dioxide enters the mesophyll cells and combines with the phosphoenol pyruvate, a 3-carbon compound, to form a C4 acid oxaloacetic acid. Thus, reaction is catalyzed by the enzyme is phosphoenol pyruvate carboxylase. C4 pathway, the reaction occurs in the cytosol of mesophyll cells and is called fixation of carbon dioxide or carboxylation. C4 acid is transported from cytosol of mesophyll cells to bundle sheath cells of chloroplast. C4 acid is decarboxylated to release carbon dioxide near rubisco in bundle sheath cells. The other product of decarboxylation reaction, pyruvate, is transported back to mesophyll cells where it is used in regenerating phosphoenol pyruvate. C4 photosynthetic pathway is more efficient than C3 pathway due to absence of photorespiration.